Hi everyone, welcome back to our Biology 2401 lecture series. I am Dr. E. Today we are exploring Chapter 3, Cells, the Living Units. Every structure and function in the human body begins at the cellular level. So in this lecture, we'll cover how cells are organized, how they communicate, how they transport material, and how their internal structures like the nucleus, mitochondria, and plasma membrane keep the body running smoothly. By the end of this lecture, you will see that understanding cells isn't just about memorizing facts. It's about understanding how life itself operates. Just to note, we will break this lecture into two parts. This is part 1. I'll be making another video for part 2. Please stay tuned for that. So grab your notes and let's dive into the incredible world of the cell, the living unit of the human body. Before we get into the details of membranes, organelles, and cell transport, let's talk about why studying cells is so important. Every living thing, from the simplest bacterium to the most complex human, is made up of cells. Cells are the basic structural and functional units of life. That means everything your body does, from contracting a muscle to thinking a thought, starts at a cellular level. Understanding how cells work helps us understand how the human body works. When you learn about the plasma membrane, you're learning about how cells communicate and protect themselves. When you study organelles, you are uncovering how energy is produced, proteins are made, and waste is managed. And when things go wrong, for example, when a cell can't divide properly, can't transport nutrients, or can't repair DNA, that's when disease develops. For example, conditions like cancer, cystic fibrosis, and sickle cell anemia all trace back to cellular dysfunction. So studying cells is the foundation for understanding health, disease, and the incredible complexity of life itself. All right, let's now explore how these living units keep us alive and function every day, every second of the day. So what is a cell? A cell is defined as the smallest structural and functional unit of living things. In a human body, there are about 50 to 100 trillion cells. And according to the cell theory, all living organisms are made up of one or more cells. And typically, these cells arise from different cells. There is a great diversity in cell types. There are over 250 different cell types in the human body. And these cells differ in their size, shape, and subcellular components. We'll review these different cell types on the next slide. This figure illustrates the diversity in cells and highlights the different functional category of body cells. In figure A here, we see cells that connect body parts, form linings, and transport gases. For example, fibroblasts, erythrocytes or red blood cells, and epithelial cells. Figure B highlights cells that move body parts or organs. Examples include skeletal muscles and smooth muscles. Figure C shows cells that store nutrients. Examples are fat cells or adipocytes. Figure D shows cells that are involved in immune responses and fighting off invasion by pathogens, for example, macrophages. And figure E shows cells that are involved in information gathering, processing, and controlling of body functions like neurons and nerve cells. And finally, figure F shows cells that are involved in reproduction, for example, sperm. Although cells have different sizes, shapes, and functions, all cells have some common structures and functions. Human cells have three basic parts. The first part is the plasma membrane, the second is the cytoplasm, and the third part is the nucleus. We are going to review each of these parts as we go through this lecture. But very quickly, the plasma membrane is a flexible outer boundary and regulates entry and exit of material into and out of the cell. The cytoplasm is the intracellular fluid and contains the organelles. The nucleus is the control center of the cell. It contains the DNA that regulates cellular activities. This figure depicts the structure of a generalized cell. The cell is depicted as roughly oval in shape, with a plasma membrane enclosing the entire cell. As you can notice, the top of the cell is cut off to expose the interior of the cell. Within the cell are organelles. Organelles are specialized structures within the cell that perform specific function for that cell. The organelles include the nucleus, cytoplasm, lysosomes, mitochondria, endoplasmic reticulum, Golgi, and many other organelles. We will review each organelle as we go through this lecture. Now let's talk about extracellular materials. Extracellular materials are substances found outside cells and include extracellular fluids, cellular secretions, and extracellular matrix. Examples of extracellular fluids are interstitial fluids, or the fluid that cells are submerged in, blood plasma, or the fluid of the blood. So blood is made of two components, a liquid component called plasma and a solid component called the blood cells. Another example of extracellular fluid is the cerebrospinal fluid. The cerebrospinal fluid surrounds the central nervous system, that is, the brain and the spinal cord. Examples of cellular secretions include saliva, mucus, and gastric fluids. Extracellular matrix are substances that act as glue to hold cells together. We start our review of cellular components with the plasma membrane. The plasma membrane is an active barrier separating the intracellular fluid from the extracellular fluid. The intracellular fluid is the fluid within the cell. The plasma membrane plays dynamic roles in cellular activity by controlling what enters and what leaves the cell. In fact, the plasma membrane is described as selectively permeable because it regulates entry and exit of substances into and out of the cell. It is also called the cell membrane. So what makes up the plasma membrane? The fluid mosaic model has been used to describe the plasma membrane. In this model, the membrane is depicted as a thin structure composed of a double layer or a bilayer of phospholipids with proteins dispersed or embedded in the bilayer. The membrane also contains cholesterol and surface sugars that form the glycocalyx. Within the membrane are structures that hold cells together through cell junctions. Now let's review membrane lipids. Most of the lipids on the plasma membrane are phospholipids, which are composed of two parts: a phosphate head, which is polar and hydrophilic or water-loving, and a fatty acid tail, which is non-polar and hydrophobic or water-hating. Approximately 20% of membrane lipids are cholesterol. Cholesterol is located between phospholipid tails and typically plays a role in membrane fluidity. So an increase in cholesterol on the membrane increases membrane stiffness. This is an illustration of the phospholipid bilayer. Each phospholipid has a polar hydrophilic head and a non-polar hydrophobic tail. Notice that the polar heads are facing the watery intracellular and extracellular environment. Meanwhile, the hydrophobic tails are hiding within the bilayer, away from water. Let's discuss membrane proteins. A cell's plasma membrane is made up of proteins that allow it to communicate with its environment. Proteins make up about half of the plasma membrane by mass and are responsible for most of the specialized membrane functions. Some proteins float freely along the membrane surface, while others are restricted to intracellular structures. There are two distinct populations of membrane proteins: integral membrane proteins and peripheral membrane proteins. Integral proteins are firmly inserted into the lipid bilayer. Some protrude from one face of the bilayer only. Most integral proteins are called transmembrane proteins and span the entire membrane and also protrude on either sides of the membrane. All integral membrane proteins have hydrophobic and hydrophilic regions, which allow them to interact with both nonpolar phospholipid tails and the water inside and outside the cell. Some integral membrane proteins are involved in transport, forming channels and pores. Others act as carriers that bind to a substance and move it across the membrane. Other transmembrane proteins act as enzymes, while others act as receptors. Unlike integral proteins, peripheral proteins are not embedded in the lipid bilayer. Instead, they are loosely attached to the integral proteins or have a hydrophobic region that anchors them to the membrane. Some peripheral proteins function as enzymes. Others are motor proteins involved in mechanical functions such as changing cell shape during cell division and muscle contraction. Others play a role in linking cells together. This is a schematic representation of the plasma membrane. As already mentioned, the plasma membrane is a phospholipid bilayer with embedded proteins arranged as a fluid mosaic. Plasma membrane lipids include phospholipids and cholesterol. Phospholipids form the basic structure of the plasma membrane. Cholesterol stiffens the membrane and decreases water solubility. Proteins determine what function the membrane can perform and could be integral or peripheral. The cell membrane also has carbohydrates, which are involved in identification of molecules and allow for recognition of self versus non-self. Carbohydrates on the outside of the cell forms a coating called the glycocalyx. What are the functions of membrane proteins? This figure summarizes the main functions of membrane proteins. I will highlight just a few. Figure A shows how membrane proteins function in transport. Transport proteins allow cells to control which substances enter or leave the cell. Figure B shows how membrane proteins function as receptors for signal transduction. Receptors allow a cell to receive information from other cells. A receptor's extracellular binding site fits the shape of a specific chemical messenger, such as a hormone. Figure C shows how membrane proteins function in enzymatic activity. Enzymes in the membrane allow cells to transform one substance to another. Figure D shows how membrane proteins function in cell recognition. Some proteins in the membrane of adjacent cells allow them to recognize each other through direct physical contact. Next, we look at the glycocalyx. The glycocalyx consists of sugars or carbohydrates sticking out of the cell surface. Some of the sugars are attached to lipids, in which case they are called glycolipids. Others are attached to proteins, in which case they are called glycoproteins. Every cell type has different pattern of the sugar coatings. They function as specific biological markers for cell-cell recognition. They also allow the immune system to be able to recognize self from non-self. Next, we look at intercellular junctions. Intercellular junctions allow adjacent cells to adhere or communicate. There are three types of intercellular junctions: tight junctions, desmosomes, and gap junctions. Tight junctions are impermeable cell junctions that form continuous seals around the cell to prevent molecules from passing between cells. A tight junction is formed by a series of interlocking junctions and has space between the cells. Desmosomes are anchoring junctions that bind adjacent cells together. Desmosomes help keep cells from tearing apart. On the cytoplasmic phase of each plasma membrane is a button-like thickening called a plaque, indicated here in yellow. Thin linker proteins called cadherins extend from the plaques and fit into each other. Thicker keratin filaments, specifically intermediate filaments, extend from a plaque on one side to the plaque on the opposite side. In this way, desmosomes form an internal tension-reducing network of fibers. Gap junctions are communicating cell junctions that allow small molecules and ions to pass through. They are particularly important for communication in heart cells and embryonic cells. Hollow cylinders called connexons connect the plasma membrane of adjacent cells, forming channels through which substances can pass. Next, we turn our attention to membrane transport. Many substances must constantly move across the membrane. Some molecules pass through easily, some do not. Two essential ways substances cross the membrane include passive transport and active transport. Passive transport mechanisms do not require energy. Active transport mechanisms require energy in the form of ATP. We start with passive transport. Passive transport requires no energy input, and all types of passive transport require diffusion. Diffusion is the movement of substances from a region of high concentration to a region of low concentration. This is also described as movement down a concentration gradient. Three types of passive transport: simple diffusion, facilitated diffusion, and osmosis. This figure illustrates diffusion in 3 steps. In the first step, a dye pellet is placed in a beaker of water. The dye pellet dissolves, creating a high concentration of dye around it. In the second step, the dye molecules from the pellet are diffusing into the surrounding water. Kinetic energy makes the dye molecules move and collide constantly. As a result, they move down their concentration gradient from a region of high concentration to a region of low concentration. In the third step, the molecules from the pellet have finished diffusing and are equally distributed in the solution. When the dye is evenly distributed, there is no net movement of dye because there is no more concentration gradient. The rate of diffusion is influenced by 3 key factors. The first one is concentration gradient. The greater the difference in concentration between 2 areas, the faster the rate of diffusion. The next is molecular size. Smaller molecules diffuse faster than larger molecules, and that's because they have greater kinetic energy than larger molecules. The third is temperature. Higher temperatures increase kinetic energy, which result in faster diffusion rates. The system reaches equilibrium when there is no net movement of molecules in one direction. As already mentioned, the plasma membrane is described as selectively or differentially permeable. This is because it allows nutrients to enter the cell but prevents many undesirable substances from entering the cell. The plasma membrane also keeps valuable cell proteins and other necessary substances in the cell but allows waste to leave. Basically, it regulates what enters or leaves the cell. The hydrophobic core of the plasma membrane stops diffusion and creates concentration gradients by acting as selectively permeable barriers. Lipid-soluble molecules and small molecules can cross the plasma membrane unassisted. This is called simple diffusion. Meanwhile, water-soluble molecules and large molecules require assistance crossing the plasma membrane. This is called facilitated diffusion. Movement of water across the plasma membrane is called osmosis. So what happens if the plasma membrane is severely damaged? Severe damage to the plasma membrane results in substances diffusing freely into and out of the cell, thereby compromising concentration gradients across the membrane. For example, patients suffering from burns lose fluids, proteins, and ions from damaged cells. These patients are at increased risk of dehydration. Let's review simple diffusion. In simple diffusion, substances diffuse directly through the lipid bilayer unassisted. Non-polar lipid-soluble substances or hydrophobic substances directly cross the lipid bilayer. For example, oxygen, carbon dioxide, steroid hormones, and fatty acids would cross the lipid bilayer relatively easily. In addition, small amount of very small polar molecules like water can pass through the lipid bilayer unassisted. This figure shows simple diffusion in which lipid-soluble molecules directly diffuse through the lipid bilayer. Lipid-soluble solutes from the extracellular fluid move through the plasma membrane into the cytoplasm. The molecules dissolve in and diffuse through the lipid bilayer relatively easily. Next, we look at facilitated diffusion. This is transport across the plasma membrane with assistance of carriers or channel proteins. Facilitated diffusion can be divided into two types: carrier-mediated facilitated diffusion and channel-mediated facilitated diffusion. In carrier-mediated facilitated diffusion, substances bind to and are transported across the membrane by protein carriers. Carriers are transmembrane integral proteins that transport specific polar molecules such as sugars and amino acids that are too large for membrane channels. Binding of a molecule causes a carrier to envelop it and change shape. That results in a molecule being moved across the membrane. Carriers can be saturated when all the carriers are bound to the molecule being transported. In channel-mediated facilitated diffusion, substances are transported across the membrane through water-filled channels. Channels are transmembrane proteins with water-filled cores that transport molecules such as ions down their concentration gradient. Channels are specific based on pore size and/or charge. Two types of channels include leakage channels and gated channels. Leakage channels are always open and allow substances to leak into or out of the cell down their concentration gradient. Gated channels are controlled by chemicals or electrical signals. This figure shows two types of facilitated diffusion. In figure B, lipid-insoluble solutes from the extracellular fluid move through the plasma membrane using a carrier protein specific for that molecule. Binding of the solute causes the carrier to change shape as indicated here, which facilitates its transport into the cell. Figure C shows channel-mediated facilitated diffusion through a channel protein. Small lipid-insoluble solutes, mostly ions selected on the basis of their size and charge, may diffuse through the plasma membrane in this manner. Next, we look at osmosis. Osmosis is the movement of water through selectively permeable membranes. It's important in determining the distribution of water across various fluid-filled compartments in the body. Water crosses the plasma membrane through two main mechanisms. One is through simple diffusion across the lipid bilayer. Please note that although water is polar, it's able to cross the lipid bilayer. The second mechanism is through special channels called aquaporins. Osmosis is important in balancing the solute concentration across membranes. It occurs whenever the solute concentration differs on two sides of a membrane. This figure illustrates osmosis. Notice that water molecules can cross the plasma membrane through a specific channel called aquaporins or directly through the lipid bilayer. Next, we review osmolarity. Osmolarity is the total concentration of all solid particles in a solution. Typically, water concentration varies with the number of solid particles in that solution. So when solute concentration goes up, water concentration typically goes down and vice versa. Water would typically move from a region of low solute to a region of high solute concentration. That is to say, water moves from a region of high water concentration to a region of low water concentration. Osmosis occurs in an attempt to balance the osmolarity on either sides of a membrane. When solutions of different osmolarities are separated by a membrane permeable to all molecules, diffusion of solutes and osmosis occurs until equilibrium is reached. At equilibrium, there is an equal concentration of solutes and water across the membrane. This figure shows a U-shaped glass tubing containing two different solutions. The left compartment of the tube contains a solution with lower osmolarity or lower solute concentration. The right compartment of the tube contains a sugar solution with higher osmolarity. The two solutions are separated by a semi-permeable membrane that's freely permeable to both solutes and water. The solutes and water move down their concentration gradient in opposite directions. At equilibrium, the resulting solutions on each side of the membrane have the same osmolarity, and the volume of the solution in each compartment is unchanged. When solutions of different osmolarities are separated by a membrane that is permeable only to water, only osmosis will occur until equilibrium is reached. Water will have net movement across the membrane until osmolarity is balanced on both sides. This results in volume changes on both sides of the membrane. The volume of the side with lower solute concentration will decrease, while the side with higher solute concentration will increase. This figure here illustrates a U-shaped glass tube containing two solutions. The left compartment of the tube contains a solution with lower osmolarity or lower solute concentration. The right compartment of the tube contains a sugar solution with higher osmolarity. The two solutions are separated by a selectively permeable membrane. The membrane is only permeable to water but impermeable to solutes. So water but not solutes can move through the membrane. Water moves by osmosis from an area of lower solute concentration to an area of higher solute concentration. As a result, both sides have the same osmolarity but different volumes at equilibrium. Next, we look at tonicity. Tonicity is the ability of a solution to change the shape of a cell or the plasma membrane tension of a cell by altering its internal water volume. We would review three types of solutions: isotonic solutions, hypertonic solutions, and hypotonic solutions. Cells placed in any of these solutions can alter their shape based on the concentration of the solution. An isotonic solution has the same osmolarity as inside the cell, so the cell retains its normal size and shape with an unchanged volume. Water moves into and out of the cell with no net gain or loss of water. A hypertonic solution has a higher osmolarity than inside the cell, so water flows out of the cell, resulting in the cell shrinking. There is a net loss of water from the cell. Shrinking is also referred to as crenation. A hypotonic solution has a lower osmolarity than inside the cell, so water flows into the cell, resulting in cell swelling. There is a net entry of water into the cell. This can lead to the cell bursting, also referred to as lysing. Next, we turn our attention to active transport. Active transport requires energy in the form of ATP to move substances across the membrane against their concentration gradient. That is to say, movement of substances from a region of lower concentration to a region of higher concentration, which is different from what we saw in diffusion. Two major active membrane transport processes include active transport and vesicular transport. We are going to review both of them. And just to know that both require ATP. Active transport can be divided into primary active transport or secondary active transport. In primary active transport, energy to do work comes directly from hydrolysis of ATP by transport proteins called pumps. Energy from hydrolysis of ATP causes change in shape of transport protein, which causes a solute such as ions bound to the protein to be pumped across the membrane. An example of such pumps is a sodium-potassium pump or sodium-potassium ATPase on the cell membrane. As already mentioned, the sodium-potassium pump or sodium-potassium ATPase is an example of a primary active transport process. The pump is an enzyme that hydrolyzes ATP to release energy required to move ions across the membrane against their concentration gradient. 3 sodium ions are pumped out while 2 potassium ions are pumped into the cell. This figure shows the role of this pump in 6 steps. In step 1, 3 cytoplasmic sodium ions bind to the pump protein on the cell membrane. In step 2, sodium binding promotes hydrolysis of ATP. This reaction releases energy, which phosphorylates the sodium-potassium pump. In step 3, phosphorylation causes the pump to change shape. The change in shape pushes the sodium out of the cell. In step 4, 2 extracellular potassium ions bind to the sodium-potassium pump. In step 5, the binding of the potassium ions triggers release of the phosphate. Once the pump is dephosphorylated, it resumes its original shape or conformation. In step 6, the pump protein binds ATP and releases potassium into the cytoplasm. The pump's binding sites are now ready to bind sodium ions again, so the cycle can be repeated. The sodium-potassium pump is essential in maintaining the electrochemical gradient across the membrane, which involves both the concentration and electrical charge of ions. This is critical for the function of muscles and nerve tissues. Next, we discuss secondary active transport. In secondary active transport, energy stored in concentration gradient of ions created by primary active transport pumps is used to move substances across the membrane against their concentration gradient. Secondary active transport always moves more than one substance at a time using a co-transport protein. Secondary active transport can be divided into 2 types, symport and antiport. Symporters transport 2 different substances in the same direction. Antiporters transport one substance into the cell while transporting a different substance out of the cell. Some sugars, amino acids, and ions are usually transported into the cell via secondary active transport. This figure illustrates secondary active transport using the ATP-driven sodium-potassium pump as an example. Note that sodium ions are more concentrated outside the cell and potassium ions are more concentrated inside the cell. In the first step, which is a primary active transport process which we've discussed already, the ATP-driven sodium-potassium pump stores energy by creating a steep concentration gradient for sodium entry into the cell. The pump drives sodium ions out of the cell and pumps potassium back into the cell. In the second step, which is a secondary active transport mechanism, sodium ions diffuse back across the membrane through a membrane co-transport protein. This drives glucose against its concentration gradient into the cell. The sodium-glucose symporter loads glucose from the extracellular fluid and moves it across the membrane. Then, the sodium-glucose symporter releases the glucose into the cell cytoplasm. Our next topic is vesicular transport. Vesicular transport moves large substances across membranes in structures called vesicles. Because it is an active transport mechanism, it requires energy in the form of ATP. We will focus our attention on 2 main types of vesicular transport, endocytosis and exocytosis. Endocytosis is transport into the cell, and there are three types that we'll talk about: phagocytosis, pinocytosis, and receptor-mediated endocytosis. Exocytosis is transport out of the cell. Endocytosis is transport into the cell and involves formation of protein-coated vesicles. It usually involves receptors and therefore can be a very selective process. So substances being pulled into the cell must be able to bind to their unique receptors. Unfortunately, some pathogens are able to hijack the receptors for transport into the cell. Once a vesicle is pulled into the cell, it may fuse with a lysosome or undergo transcytosis. This figure illustrates 6 steps in endocytosis. In step 1, a protein-coated pit in the cell's plasma membrane ingests a substance. In step 2, a protein-coated vesicle detaches from the pit and into the cell. It loses its protein coat as it moves further into the cell. In step 3, the coat proteins are recycled back to the cell's plasma membrane. In step 4, the vesicle is now an uncoated endocytic vesicle. It fuses with a sorting vesicle called an endosome. In step 5, a transport vesicle may carry some of the membrane components of the fused vesicle back to the cell membrane for recycling. In step 6, the fused vesicle may either A. fuse with the lysosome for digestion of its content, or B. deliver its content to the plasma membrane on the opposite side of the cell. If the vesicle's contents are delivered to the opposite side of the cell, this is known as transcytosis. Next, we review the different types of endocytosis. We start with phagocytosis. Phagocytosis is also referred to as cell eating. In this process, the cell engulfs a large particle by forming projecting pseudopods around the particle. The term pseudopod means false foot. The pseudopods enclose the particle within a membrane sac called a phagosome. The phagosome combines with a lysosome and its contents are digested. The vesicle has receptors capable of binding to microorganisms and solid particles. Next is pinocytosis. Pinocytosis is referred to as cell drinking. The cell takes in drops of extracellular fluid containing solutes. The extracellular fluid enters tiny vesicles. No receptors are used or required in this process, so pinocytosis is non-specific. Pinocytosis is used by some cells to sample their environment. It is also the main way in which nutrients are absorbed in the small intestine. Next is receptor-mediated endocytosis. In this process, extracellular substances bind to specific receptor proteins in the cell's plasma membrane. This process enables the cell to ingest and concentrate specific substances called ligands in protein-coated vesicles. Ligands may simply be released inside the cell, or they may combine with the lysosome to digest their contents. Receptors are recycled back to the plasma membrane in vesicles. Substances taken up by receptor-mediated endocytosis include enzymes, hormones, and some pathogens. The second type of vesicular transport is exocytosis. Exocytosis is a process of ejecting material from the cell. The substances being ejected are usually packaged in secretory vesicles. The vesicles contain proteins called V-snares, while the cell membrane has proteins called T-snares. The two proteins interact and trigger exocytosis. Substances removed from the cell by exocytosis include hormones, neurotransmitters, mucus, and cellular waste. This figure illustrates the 4 key steps in exocytosis. In step 1, a membrane-bound vesicle is shown in the cell's cytoplasm. The vesicle contains molecules to be secreted from the cell. The vesicle surface has membrane proteins called V-snares. This vesicle is migrating with its content to the plasma membrane. In step 2, the secretive vesicle is at the plasma membrane. The proteins on the surface called V-snares bind with membrane proteins called T-snares. In step 3, the vesicle and the plasma membrane fuse, forming a fusion pore that opens into the extracellular fluid. In step 4, the vesicle contents are released to the cell exterior through the fusion pore. Now we turn our attention to membrane potential. As we are aware, the plasma membrane is selectively permeable. The permeability of the plasma membrane results in generation of a membrane potential or a voltage across the membrane. At rest, all cell membranes have electrical potential energy produced by separation of oppositely charged particles across the membrane. The potential typically ranges from minus 50 millivolts to minus 90 millivolts, depending on the cell type. For this reason, all cells are said to be polarized. The minus sign before the voltage indicates that the inside of the cell is negative compared to its outside. The voltage or charge separation exists only at the membrane. While all body cells have membrane potential, it is especially important for nerve cells and muscle cells because they use changes in membrane permeability as a form of communication. So how is the resting membrane potential generated? There are many kinds of ions found inside and outside the cell. However, the resting membrane potential is determined mainly by the concentration gradient of potassium ions and by the differential permeability of the plasma membrane to potassium. The cell cytoplasm contains more potassium ions than the extracellular fluid. The cytoplasm also contains protein anions that are unable to follow potassium through the plasma membrane. As you can see from this diagram, the inner face of the plasma membrane has a negative charge, while the outer face of the plasma membrane has a positive charge. The figure shows 3 key steps in generating the resting membrane potential. In step 1, potassium ions diffuse down their steep concentration gradient out of the cell via leakage channels. This loss of potassium ions results in a negative charge on the inner face of the plasma membrane. In step 2, potassium ions also move into the cell because they are attracted to the negative charge established on the inner face of the plasma membrane. In step 3, when the movement of potassium ions out of the cell equals the movement of potassium ions into the cell, a negative membrane potential of negative 90 millivolts is established. At this point, the concentration gradient promoting the exit of potassium ions exactly opposes the electrical gradient for the entry of potassium into the cell. As we just discussed, diffusion establishes the resting membrane potential. So how is the resting membrane potential maintained? Active transport processes maintain the membrane potential to keep it in a steady state. This requires that the rate of active transport of sodium ions out of the cell is equal to and dependent on the rate of sodium diffusion into the cell. The sodium-potassium pump actively pumps 3 sodium ions out of the cell and pumps back 2 potassium ions into the cell. The sodium-potassium pump maintains both the concentration of ions and charge distribution across the membrane. This makes up the electrochemical gradient across the membrane. The transient opening of gated sodium and potassium ions in the membrane changes the potential from its resting value. This is a normal means of activating neurons and muscles. We will discuss this topic in more details when we get to the nervous system. Our next topic of discussion is how cell adhesion molecules and membrane receptors allow the cell to interact with its environment. Cells interact with their environment by responding directly to other cells or indirectly to extracellular chemicals. These interactions always involve the glycocalyx. The most important molecules cells use to interact with the environment fall into two large families, namely cell adhesion molecules and plasma membrane receptors. What are the functions of cell adhesion molecules? Thousands of cellular adhesion molecules, also called CAMs, are found on almost every cell in the body. CAMs play key roles in embryonic development and wound repair, especially in situations where cell mobility is important. They also have a role in immunity. CAMs are sticky glycoproteins and perform the following functions. They anchor cells to extracellular matrix or to each other. They assist in movement of cells past one another. CAMs also attract white blood cells to injured or infected areas. CAMs stimulate synthesis or degradation of adhesive membrane proteins, such as tight junctions. They also transmit intracellular signals to direct cell migration, proliferation, and specialization. What roles do receptors play on the cell membrane? Membrane receptors are integral proteins that serve as binding sites for several chemical signals. Some function in contact signaling, while others function in chemical signaling. In contact signaling, cells touch and recognize each other by each cell's unique surface receptors. It is particularly important in normal development and immunity. Some bacteria and other infectious agents use contact signaling to identify their preferred target tissues. In chemical signaling, a ligand or chemical messenger binds to a specific receptor and initiates a response. Examples of ligands include neurotransmitters, hormones, and paracrines. Different cells respond differently to the same ligand. The question is why? The answer is that the target cell's response depends on the internal machinery that the receptor is linked to, not the ligand. While responses differ from cell to cell, ligand binding typically induces a change in the shape of the receptor that becomes activated. For example, some membrane receptors respond to ligands by becoming activated enzymes, while others in muscles and nerves respond by transiently opening or closing ion channels, causing changes in excitability. All right, that brings us to the end of chapter 3, part 1. Thanks for sticking around. Please stay tuned for part 2 coming out soon. Bye for now.